Good evening. Today we will be discussing the microstructure or the histology of the cerebrum and the cerebellum. We have already done the ganglion and the spinal cord, peripheral nerves. So let's discuss the cerebrum and the cerebellum histology. You now know the central, we have divided the nervous system into two main parts, that is central nervous system and the peripheral nervous system. Brain and the spinal cord, they are part of the central nervous system. Peripheral nervous system has peripheral nerves, cranial nerves, spinal, and the spinal nerves and the cranial nerves, as well as includes the ganglia. Ganglions, the sensory ganglion, which are the dorsal root ganglion, we have cranial nerve ganglions, sympathetic ganglion, parasympathetic ganglion, they all which are developing from the neural crest. You've done the spinal cord, in which we saw that the gray matter was lying inside and the white matter was outside. While now, when we see the slide of the cerebrum and cerebellum, we have the cortex, which is the gray matter, which is lying outside both in cerebrum as well as in the cerebellum. Now, what is the reason why from the spinal cord the gray matter has come outside and the white matter is inside, while in the spinal cord it was the opposite. The gray matter was inside and the white matter was outside. This has a developmental reason, that this is a neural tube which we are seeing here. The neural tube, when it has the alar lamina and the basal lamina, when we've seen in the spinal cord, the alar lamina, which was giving to the posterior horn, and the basal lamina, which is giving rise to the anterior horn, motor and the sensory. And this, the cavity of the brain was the central canal. As when we reach the cerebellum and the cerebrum, which is the developing from the prosencephalon, from the telencephalic vesicle, while the cerebellum is part of the brainstem from the rhombic lip. It develops from the rhombic lip, and the lining epithelium of the cavity, the roof of the cavity gets stretched, and the part of the alar lamina, which is the sensory part, it overlaps the roof, which is called as from the dorsolateral part, which is called as the rhombic lip. And from the lining of epithelial cells, the cells, they start regenerating, or the cells, they migrate towards the periphery. So that's how we get the gray matter outside and the white matter inside. But in the white matter itself, there are a few gray matter nuclei like basal ganglion, and in the cerebellum, you've done the dentate nucleus, the emboliform, the fastigious. They were lying inside the white matter. They fail to migrate. Those neurons which fail to migrate, they are lying in the white matter, deep cerebellar nuclei. That's how the cortex has gone outside and the white matter is inside. So we are seeing opposite of the spinal cord. Because of the migration of the neurons from the inside to outside, from the epithelial lining, which is ependymal lining, the central canal, and the cavity of the cerebellum is the fourth ventricle. So we have, that's can be explained that this is why the gray matter is outside. We need more of the gray matter for control. The cerebellum is controlling the vasomotor activity of the whole body. So we have number of folias also. This is just showing the growth of the ependymal, the growth which is giving rise to the epithelial lining, which will give rise to the neurons, and this is the pontine fissure. So this part from here, the fourth ventricle, and from the rhombic lip will develop the cerebellum. Now coming to the histology of cerebral cortex. The cerebral hemispheres consists of convoluted cortex of the grey matter overlying the central medullary mass or the white matter, which has got the myelinated fibers, and the grey matter consists of neurons, cell body of the neurons, their dendrites, and interneurons plus the neuroglial cells. The white matter conveys the fibers between the different parts of the cortex and brings the information from the other parts of the cortex. So it is the cerebrum or the cortex is connected to the other parts of the brain as well. Now, what are the layers of the cortex? If you see, this is a picture showing the brain, where you are seeing this white thing is the white matter, and inside 2/3 of these are the gyri and the depressions are the sulci. So 2/3 part of the sulcus is lined with the grey matter cortex, and the rest, this is the white matter, and these are the grey matter nuclei, which are the thalamus and the caudate nucleus. Now here you are seeing this as the part of the cortex, and this is the white matter. This is one folia. Not folia in this, but the sulcus, and this is the elevations are the gyri. Now what are the cell types or the cells you will be seeing in the cerebral cortex? These are the most, in almost all the layers we have the distribution of the pyramidal cells of different sizes of pyramidal cells. Small pyramidal cells, medium or the moderate pyramidal cells, and the large pyramidal cells. These are the motor cells. Then we have the stellate cells. Two principal cell types are the pyramidal and the stellate cells. The other cells are the cells of Martinotti and fusiform cells, which resemble the pyramidal cells. The others in the superficial layer of the cortex are the horizontal cells of the Cajal. The cells which you are seeing, this is a pyramidal cell having a base and the apex. From the apex starts the dendrite, and from the base is the axon. And these are the large pyramidal cells which are present in the fifth layer of the cerebrum, which are also called, this layer is also called as the ganglionic layer, because of the presence of the larger, more concentration of the large pyramidal cells. Then we have the cells, this is the cells of the Martinotti, which is the pleomorphic layer, which are present also, the axons of these, they travel to the superficial layer and divide in a T-shaped manner. Then these are the medium size pyramidal cells, and this is the fusiform. The shape is like a spindle or a fusiform, and this is they do not go out of their dendrite processes do not go out of the brain or lie itself in the cortex. Then we have the small pyramidal cells, and these are one of the neuroglial cells, and the other cells which are present is the basket cells and the stellate cells. These are the small pyramidal cells. So the blue represents the afferent fibers, and the red is the efferent fibers, and those which are shown as the black, they are limited to the cortex itself. I hope this is clear. So let's move on to their individual characteristics of the cells. The pyramidal cells are pyramidal in shape, as the name suggests. We're having a base and the apex. They are the largest of the neurons present in the cerebrum, around 70 micron in diameter. These are pyramidal cells, and from the apex arising is the dendrite, and from the base we have the axon. The largest of these pyramidal cells which are present in the motor cortex. Now you are familiar with the motor cortex, that is area 468. So we have a ganglionic layer where the large pyramidal cells are called as the cells of the Bex. Now what are the functions of these pyramidal cells? They have apical dendrites, and their dendrites are studied with dendritic spines which make synaptic connections, and these dendrites are like small projections on the dendritic processes where the synapses, these are these sites of synapses that are selectively modified as a result of learning. Most of all pyramidal cells have long axons and leave the cortex to reach the other cortical areas or the various subcortical sites. Therefore, the principal output of the cerebral cortex is from the pyramidal cells. Now, the other principal cell which we discussed was the stellate cells. They are principally the interneurons and are of different sizes and shape. They are multipolar, and their function is that these stellate cells are the principal interneurons present in the neocortex. They do not leave the cortex. They are limited to the cortex of the cerebrum. Then the cells of the Martinotti, the fancy name, and they are more polygonal in shape and have very short dendrites. The axons, they bifurcate superficially, they come to the horizontal layer, they bifurcate horizontally in a T-shaped manner in the superficial layer of the cerebral cortex, and also form synapses with the pyramidal cells. Fusiform cells, they are spindle shaped, and their function is similar to the pyramidal cells. Their dendrites extend, they are deeper and more into go into the superficial layers. The other cells which you will see in the superficial layer, that is plexiform layer. We have 6 layers in the neocortex. So these cells, horizontal cells of Cajal or Cajal cells, they are small spindle shape oriented parallel to the surface. They are present parallel to the surface and are less common type. Why? Because they disappear after birth, and 1 or 2 cells are left. Found only in the superficial layer. Axons pass laterally to synapse with they are also going to form synapse with the pyramidal cells. Present in the layer, mainly present in the plexiform layers. Now, if this is the slide of the cerebrum, which is showing the coverings. So any slide which you see, you will see the coverings of the meninges. So it is showing the arachnoid matter, and this is the pia matter, and deep to it is the external limiting membrane. And you can see this is lighter stained as compared to the deeper part. So this layer is primarily has got the fibers coming from the deeper layers, and either the axons or the dendritic processes which form plexiform network between these fiber synapses, and the nuclei which you are seeing are of the neuroglial cells. Here you can see the laminated arrangement. Why does the cerebrum appears laminated? It has different shapes and sizes of the neurons and are arranged vertically and radially, which give it a laminated appearance. So when you draw, you have to draw the biomatter as well as the coverings of the cerebrum of the cortex. Now, what are the different types of cortex? We have neocortex, which is a 6-layer cortex, and it is a homogenic cortex. Then paleocortex, which covers some parts of the brain, that is the olfactory area, and forms heterogeneous cortex. That means there are different types of mainly fibers and cells present. Then the archicortex, like you've done the archicerebellum, archicortex is the oldest of all, and it is present in the hippocampal formation, archicortex. This is the newer appearance of the cortex, and paleo is in between new and archi. Now the axons are arranged in horizontal and radial pattern. Axons are in the plexiform layer horizontally, and few here are going in the radial pattern in all layers, giving a laminated appearance to the cortex. You can see these different laminas. These are actually different layers of the cerebral cortex. Coming to the 6-layer cortex, which is the neocortex. The outermost is the molecular layer. Then comes the outer granular layer, and then the inner outer pyramidal cell layer. The outer pyramidal cells layer has got smallest of the pyramidal cells. So plexiform, outer granular, and then outer pyramidal. After the outer pyramidal, we have the inner granular layer. We'll come to the cells and how to differentiate. Inner granular layer, which has more number of granule cells or the stellate cells. Then the pyramidal layer or the ganglionic layer, which has the largest of the pyramidal cells. Then the multiform layer. So out of these neocortex, we have a granular cortex and agranular cortex. The granular cortex is mainly the sensory cortex, like area 312, auditory areas, visual areas. They have the granular cortex. That pyramidal layer is less prominent as compared to the granular layers. And the agranular cortex or the granular cortex, agranular cortex is the motor cortex. This is to show you, this is the molecular layer, lightly stained, outer granular layer, which has got the stellate cells, granule cells, and outer pyramidal layer, which have got the small pyramidal cells, inner granular layer. So in between, first you remember molecular, outer granular and outer pyramidal. So now again we have inner granular, inner pyramidal and pleomorphic. So that is how you can remember these layers. The outermost molecular layer, external granular layer, external pyramidal layer, outer pyramidal or external pyramidal, one and the same thing. This is the H&E stain where you can see the molecular layer is the plexiform layer. Another name for it is the plexiform layer. Then comes the external granular and external pyramidal layer number 3, which has small and medium sized pyramidal cells. Then the inner granular, which has horizontal fibers also running here, and this is called as the outer band of Belliager. The 5th one is the ganglionic, large pyramidal cells. So large pyramidal cells, medium sized pyramidal cells and small pyramidal cells. The small pyramidal cells, they connect the association between the two areas within the cerebrum. The medium size, they connect the same areas into cerebral hemispheres, and the large number of fibers which forms the longitudinal fasciculus. They connect longitudinally, the fibers run from one cerebral hemisphere to the other hemispheres. Now, this is what are the components of the plexiform layer. It contains the dendritic and axonal synapses, with one another, and there are very few nuclei seen which belong to neuroglia, and occasional present cells, neurons, is the horizontal cells of the Cajal. This is how the first three layers will look like. This is the nerve fibers present in the what are the components of the plexiform layer? Synapses and the axons and the dendrites, horizontal cells of Cajal, and these nuclei are nuclei of the neuroglial cells. So this is showing the components of the plexiform layer. Outer granular, you have seen dense population of small pyramidal cells and stellate cells, and also contains exodendritic synapses. So next, what are the components of the inner granular layer? We have the neuroglial form cells, stellate cells, and smallest pyramidal cells in the outer granular layer. They are densely packed. Why it appears more blue is because of the nuclei of the stellate cells, which are densely packed. And we have also is the outer band of belly agar, which is present in the third layer. So that is formed is the third layer, which is the pyramidal cell layer. Moderate size pyramidal cells predominate. Large pyramidal cells are present in the further deeper cells. Cells of Martinotti may be present in the pyramidal cell layer. What are the components of this? We will find few of the fusiform and basket cells, plus small and medium sized pyramidal cells. So in the first layer, it was the plexiform, second was the outer granular layer, and third one is the pyramidal cell layer, which has got the small pyramidal cells. The fourth is again the inner granular layer. I have missed out on that because the same features as the outer granular layer, but the next fifth is called as the ganglionic layer, or the cells of bed. Here large pyramidal cells are present, stellate cells, cells of Martinotti, and largest of the pyramidal cells, also called as these Bett's cells, are present in the motor cortex area 4, 6 and 8. And because they are larger, hence the name given to them is the ganglionic cell layer. You can see for the comparison, we've seen this is the plexiform, outer granular, and then the pyramidal cell layer. This is the inner granular layer, fourth layer, and this is the fifth is the inner pyramidal cell layer and pleomorphic layer. So just to compare, these are the first three layers and these are the next three layers. So we have the inner granular, pyramidal cell layer or the ganglionic layer, and this is the pleomorphic layer. and then comes the white matter. Is it clear to all of you? I hope. The 6 layers. Now, what are the components of the ganglionic cell layer? We have few cells of stellate cells and cells of Martinotti, which also make synapses with the pyramidal cells, and it shows the inner band of belly agar. The outer band of belly agar is present in the inner granular layer, and the inner band of belly agar, that is the white band, white matter band, which you see is present in these 2 layers. Fourth one, granular. Inner granular has external band of belly agar. The 5th layer or the ganglionic cell layer has got the inner band of belly agar. This I will come to, and large pyramidal cells are present, including the cells of the beds. So you can see, even you can differentiate in this lower magnification, the cells nuclei are much larger as compared to this granular and the 3rd layer. So let's move on to the multiform layer, which has got all the morphological forms found in this layer, and the fusiform cells are present in the deeper part. Other cells are present in the superficially in this layer, other cells. The additional features which you will see in the visual cortex is called as the stria of Gineri. This is prominent external band of belly agar, which is present in the lamina 4 or the inner granular layer. So we have the stria of Gineri, which can be seen in the visual cortex or the striate cortex. Another name is the granular cortex. This is seen in the lamina 4. This is called as the stria of Gineri. And the maximum density of the stellate cells, that is a granular layer. So the granular layer has got maximum density of stellate cells, which is called as the coniocortex. There are different types of cortex which you can come across. We have the agranular cortex, where the 2nd and the 4th layer almost absent, and area 4, 6, 8, that is motor area, and the limbic cortex has got the agranular cortex. That means the granular layers are almost disappearing or absent, and it has densely packed pyramidal cells. The other important cortex, so if you are able to differentiate at your level, agranular and granular cortex is good enough. We have the almost absent 3rd and 5th layer. That means pyramidal cell layer is less, large pyramidal cells thickness is less, densely packed stellate cells. Here they are densely packed pyramidal cells. Motor cortex, this is the example of the sensory cortex. Area 312, visual area 17, 18, 19, auditory area 41, 42, and parahippocampal gyrus. So all the sensory cortex has got the granular cortex. Motor cortex is agranular cortex. Then they are on the frontal parietal poles, lobes. We have the prominently reduced 3rd and 5th layer. The prominent layers are the 3rd and 5th frontal cortex, again the motor cortex, and reduced 2nd and 4th, that means outer granular and inner granular, reduced. Parietal cortex, prominent 2nd and 4th, means granular layers, and reduced 3rd and 5th means pyramidal cell, they are parietal lobe. Then there another name given is all layers are diminished at the poles of the lobes, that is frontal and occipital pole. This is just to show you again these fusiform cells, how they look like in Golgi stain. Special stains, when they do, you can see the dendritic processes as well as the axonal processes. So horizontal cells which make synapses with the dendrites of the pyramidal cells. Then the cells of Martinotti, they also make synapses with the pyramidal cells. And the stellate cells, which are stellate cells, have got synapses with the afferent fibers coming, and the pyramidal cells also have got dendritic synapses with the afferent fibers. So these represents the efferent fibers, and this is the efferent fibers present in the white matter, cerebellum. Now, what are the afferent inputs? We have the association nuclei coming from the thalamocortical, all the fibers coming from different areas are coming to the cortex are the afferents. And efferent is corticothalamic, cortical, corticostriate, basal ganglia, and fibers going from cortex to spinal cord and brainstem, corticospinal, corticonuclear, corticopontine, all these are the efferent fibers. Now, you must be knowing more than me now. So this is just to revise again. The first layer, molecular layer, granular layer. So two external layers, external granular, external pyramidal. Internal granular, internal pyramidal, or the ganglionic. That is how you can remember. And the odd one out is the pleomorphic or the polymorphic layer, which has got all the different sizes and shapes. This is just to say the components of the molecular layer. Molecular layer, neuroglia and horizontal cells, fibers of neuroglia, small pyramidal neuroglia, external pyramidal layer, third layer, medium sized pyramidal cells, granule cells. This I have already done. You can revise it by revisiting this table, and that is how you will remember. The other components of the cortex are the neuroglial cells, which are of different types. We have already done astrocyte, oligodendroglia, microglia, and ependymal cells. Now, what can be the diseases associated with the cerebral cortex? Is one of most commonest is the Alzheimer's disease, which you have. They can be plaques developing and neurofibrillary tangles. And the another one is the dementia. There are neuronal inclusions, degenerative inclusions in the cerebral cortex. Then Huntington's disease, vascular dementia because of the multiple infarcts. The blood supply is because of atherosclerosis or narrowing of the blood vessels. They have developed multiple infarcts which give rise to dementia. Then of course the neoplasms, astrocytoma, oligodendrocytoma, and these other diseases which you will learn later are the most commonest for you people to remember is Alzheimer's and the dementia. This is how the cerebral infarct will look, which has got three components: the fibrous component and the cellular components, and in between these the nuclear inclusions. There can be amyloid deposition in the cortical arterioles, so cerebral, which is called as the cerebral amyloid angiopathy. Now coming to the cerebellum. Now you are familiar with the cerebellum also. So you can see these folias and the fissures of the cerebellum. This division of these white matter running inside these folias this is called as the arbor-vitae appearance. And the fibers which is forming this is a core of the cerebellum. So again the same thing, the gray matter is lying outside and the white matter is inside. Is the same pattern as that of the cerebrum to increase the surface area. This is controlling the vasomotor, unconscious sensations of the body. So we need lot of, for whole body we need lot of gray matter. So that is how the God has provided us, that outside with number of folias and fissures is the gray matter and inside is the white matter, and those neurons which fail to migrate, they are forming the deep cerebellar nuclei. So this is how the cut section, horizontal section of the cerebellum will look like. So this is the arbor-vitae appearance of the cerebellum, which is quite a peculiar thing. Just when the slide is given, you can identify this fissures and folias, and by looking at this only you can identify the slide. So whenever the nervous system slide is given, pick up the slide and see it with your naked eye. So macroscopic, half of the slides you will be able to distinguish with your naked eye. So first step is, before putting under the microscope, look at the slide from with your eyes, naked eye, and then put it in the microscope. Now there are 3 layers as come. Now less to remember. There were 6 layers in the cerebrum. Here we have only 3 layers. Again, this plexiform was now is called as the molecular layer, Purkinje cell layer, and the granular layer. And we have the molecular layer. Mother, please go to garden, or mother, my for garden. Then this is how there are mnemonics by which you can remember, but I think it's easier. Molecular layer is the which has lightly stained and less of nuclei. Then we have the Purkinje cell layer, and then we have the granular layer. There is no internal or outer granule, inner or outer granule. So you can differentiate the cerebrum, which was 6 layers in the neocerebellum, neocortex, and we have 3 to 6 layers of the neocortex. Archicortex is the older cortex. There may be only 3 layers present, and in between is the paleocortex. And cerebellum, whether it is archicerebellum, it's paleocerebellum, or neocerebellum, it is the 3-layered cortex. There is variable thickness, and this is the uniform thickness everywhere. Cerebellum has got uniform thickness. There are 5 subtypes based on the distribution, no further types. So we've divided it into agranular, granular, polar, and while here we have no further subtypes. Cortical output is largely excitatory, and here the cortical output in the cerebellum is largely inhibitory. Now, what are the different types of neurons or the cells present in the cerebellum? We have the stellate cells, the basket cells, and the granule cells, and Golgi type 2 neurons, or the Golgi type 2 cells. You can see here the basket cells, Purkinje cells, which are flask shaped, and the granule cells, these are the D is for the dendrites of the Purkinje cells. This is a section which is both this part is the cut as the longitudinal section, and this is cut as the transverse section. This is a fissure, you can see. This is part of the folia. So you can see the arrangement of the cells in the three different layers. So most prominently you can see this is a Purkinje cell layer, and this is the molecular layer, which has got parallel fibers running, and the dendritic processes of the basket cells, the granule cells, and also they have the dendritic processes of the Purkinje cells. So molecular layer, Purkinje cell layer, and the granular layer. In the granular layer, you can see just deep to the Purkinje cell layer, we have the neurons is the Golgi type, 2 neurons, and there are fibers which are ascending up and fibers which are descending down. So these are the axons of the Purkinje cells, which is the output from the cerebellum, and input is from the mossy fibers. And we have the climbing fibers, which are the only fibers coming from the accessory olivary nucleus and the main olivary complex. So the climbing fibers are the olivocerebellar, and the mossy fibers are the rest of the fibers which are coming to the cerebellum, like vestibulocerebellar, reticulocerebellar. All these are the mossy fibers. The only climbing fibers are from the olivary nucleus, that is olivocerebellar. So there are two types of fibers, olivocerebellar and the others coming from different parts. Climbing fibers and mossy fibers. And the neurons, we have said, are the granule cells, the Golgi cells, the granule cells in the molecular layer, basket cells. These all are inhibitory neurons or the interneurons in between. We can see these, there are nuclei present in these different layers, but you can see you will be seeing some islands of clear areas in between. These are nothing but called as glomerulus, which is formed by the processes of the granule cells, the axons of the Golgi cells, and these mossy fiber ends in dilatations or dilated projections. These are called as the rosette. And this whole thing with three dendritic processes of granule cells, mossy fibers, and axons of the Golgi cells, they together form a capsulated area which is called as the glomerulus, and the capsule is formed by the astrocytes around them. These are islands present in between the neurons. So what are the components of the molecular layer? The dendrites of Purkinje cells, which have gone from the Purkinje cell, which is a flask shape, from the apex is arising the dendrites. So dendritic cells of the Purkinje, dendritic processes of Purkinje cells, parallel fibers coming from the granular cells, climbing fibers, stellate cells which are present in the superficial part, and the basket cells present in the deeper part, and the neuroglial cells. These are the 6 components of the molecular layer. How does the Purkinje cell look like? Flask shape. These are single row of cells, and they have got a dendritic tree which is making synapses with the climbing fibers as well as the stellate fibers from the stellate cells. Other peculiarity is their dendritic tree is extensively placed in the molecular layer, and from the base arises the axon, which is the inhibitory pathway. Granule cells are dark staining nuclei with less cytoplasm, and they synapse with the axons of these synapse with the Purkinje cells, dendrites. Basket cells you've seen were present just deeper to the Purkinje cells, and is present in the deep part of the basket cells are present in the deep part of the molecular layer, and they course horizontally and synapse with the Purkinje cells. So the stellate cells synapsing with the Purkinje cells, the basket cells synapsing with the Purkinje cells. So they are having the inhibitory effect on the Purkinje, while stimulation, direct stimulation, is from the climbing fibers. So Golgi type 2, they are again also the inhibitory neurons with vesicular nucleus in H&E stain, and they synapse with dendrite of the granule cells, synapse with the mossy fibers. This is showing the connections of the cerebellum. So here the climbing fibers is the peculiarity of climbing fiber is that it makes only connections with the single Purkinje cell, while the effect of the mossy fibers is diffuse, and it divides the mossy fibers. This is the mossy fibers which are coming from the other brainstem nuclei. They make connections with the granule cell and the Golgi cell axon, the granule cell dendrites, the Golgi cell axon, and the mossy terminal. These three together form the glomerulus, which is encapsulated by the neuroglial cells. Here the basket cells are shown and the stellate cells, which have got inhibitory effect on the Purkinje cells, while the granule cells, they also make synaptic connections with the dendritic T of the Purkinje cells, and has got these mossy fibers has got the excitatory effect on the Purkinje cell. This is how the glomerulus look like. They are the lesser stained, light stained areas in between the nuclei of the granule cells, which are the mossy fibers, the granule cell dendrites, and the Golgi axons. This is a mossy fiber which has ended in a number of dilatations. This is called as the rosette formation, and this is the Golgi cell axon and the granule cell dendrites. They together form the glomerulus. Coming to this is showing the how this is a Purkinje cell which is getting excitatory impulses from the granule cell fibers. This is one of the granule cells which has divided into number of granule cells which have divided horizontally and are making synapses with the Purkinje dendrites. So they are excitatory to the Purkinje, while these Purkinje cell axons, when they go out, this is showing the deep nuclei. So its crying is because it is being inhibited by the axon of the Purkinje. This is a climbing fibers which, when individual gives collaterals, is excitatory, but when it comes from the Purkinje, which is Purkinje cell axon, this becomes inhibitory. So the climbing fibers, when they make synapse with the Purkinje cell bodies, then they send inhibitory impulses. So this is a mossy fiber, and this is excitatory to the granule cells. You can see this mossy granule cell axons which divide T-shaped is excitatory, and the Purkinje cell is sending inhibitory impulses to the deep cerebral nuclei. So afferents are the climbing fibers and the mossy fibers. Climbing are the olivocerebellar, and the mossy fibers are from the other brainstem nuclei. All other fibers coming to the cerebellum, cerebello-reticular, cerebello-pontine, all these are from the cranial nuclei, and they are the mossy fibers. So rest all are mossy fibers. Only afferents which are coming is climbing fibers are coming from the inferior olivary nucleus, olivocerebellar, and efferents are from the Purkinje cell axon going to the deep cerebellar nuclei, that is dentate nucleus, emboliform, vestigial, and globosus. Dentase is the lateralmost, vestigious is the medialmost. Now, how do we identify the slide of cerebellum? It is, I have shown you, its first thing you have to identify is the pia mater. Then you can see the folia and the fissures, arbor whitey appearance, outer grey cortex, inner white matter, deeply convoluted folds called cerebral folia, separated by sulci or the fissures, and white matter is forming the core of the folia. The cerebellar cortex contains only 3 layers: molecular layer, Purkinje cell layer, and the granular layer, while cerebrum had 6 layers. So outermost is the molecular layer, which contains fibers extending horizontally, and in the deeper part of the molecular layer we have the basket cells. Purkinje cell layer contains single row of flask-shaped cells. In the granular layer contains numerous small dark granule cells, Golgi type 2 neurons just below the Purkinje cells, and small clear spaces called as islands, cerebellar islands, contain the synaptic glomeruli complexes. And white matter consists of myelinated axons. This is just to revise again. This is a white matter. Then we have the blood vessels in the pia mater, and this is a pia mater, blood vessels, then the granule cells, and this is the molecular layer. Granular layer, molecular layer, small bigger dots are the Purkinje cell layer. Purkinje cell layer. Then we have the dendrites of Purkinje cells. This is the basket cells. These are the smaller islands which are showing the synaptic glomeruli. Thank you very much. I hope you have understood. Draw these diagrams, identify the layers, see the slides, and try to find out, remember these layers, because in the exam you will be asked about the rest, functional connections and all you will be doing in your physiology. Thank you for your patience. hearing. Thank you.